Hi everyone. So in this video, I'm going to go over some of the things that you may have missed the first day of lab. So if you weren't able to make it for whatever reason, I wanted to make this video. That said, this is a kind of a crash course quick video. Meant it's not going to be that quick, but it's meant to briefly go over everything. It's not meant to go over everything in great detail. So what we're going to talk about first is the lab workflow, and each lab experiment is worth 25, I should say, points. All right, so basically what's going to happen is before lab, you're going to take the pre-lab quiz on Brightspace, and at the end of this video, I'm going to go over the very first pre-lab quiz and show you what that looks like, and that is worth 10 points. During the lab, you'll start the lab most weeks with a lecture quiz. You didn't have that the very first week because you haven't covered any lecture material, but starting the second week, most weeks there is a lecture quiz, and just see the course calendar for details on when there are quizzes and when they're not, and see the announcements for what's going to be on that quiz. Then during the lab, you'll do the data collection and the analysis. You'll actually do the lab. That's the data collection part. You'll do the experiment, and then you'll analyze the data, and then you'll submit your Excel file, and that is worth 10 points. And on that Excel file, we do require that you use formulas. You can't type in the numbers. You have to use the formulas in Excel because part of this course is using the formulas in Excel. And then at the end of lab, there's a post-lab quiz. It'll ask you a few questions about exactly what you did and give you an opportunity to write a conclusion. I do want to note that Brightspace calls everything a quiz. These aren't really quizzes. These are pretty low pressure. This one you get 5 attempts. Okay, this you're uploading a file. This one is kind of more like a quiz because you only get 1 attempt, and this one is more like a quiz because you only get 1 attempt. So now I'd like to talk about what you are going to need for the lab. The first thing is you need personal protective equipment. So those are goggles, not safety glasses. Safety glasses are not acceptable. They need to be splash resistant goggles. We are working with chemicals, and if you get them in your eyes, it's big problems. Okay, a lab coat. We do prefer a cotton lab coat in certain types of lab accidents. A cotton lab coat can be much more protective because it's not as flammable as synthetic materials. Gloves, we do require the heavy duty nitrile gloves. Okay, that you're also going to need a lab manual, and the first 3 experiments are on Brightspace. Okay, so if you didn't get a chance to get a lab manual, we do have the first 3 experiments available for you in each of the folders, lab 1, 2, and 3. You can get a copy of the lab manual, but after that, we don't put them on Brightspace anymore. And all of these materials can be purchased in chemistry room B44. So that's in the chemistry building in the basement / the bottom floor. If you come from the road, it's not really the basement, it's the bottom floor. If you come from the main podium, then you go down into the basement, but it's the lowest floor you can get to, room B44. Early in the semester, there are pedestrian dividers right in the hallway, which makes it really obvious. A lot of times there's people standing out there. Keep in mind that they do carry normal business hours. They're not open late at night. It's not even really a store. It's kind of you walk in, you tell them what you need, they give it to you, and you pay for it, that kind of a thing. And basically you just need to tell them which course you're in, and if you're watching this video, you're in ACHM 115. Okay? So the next thing I want to talk about is the safety consent form. So safety is obviously our top priority. We want everyone to do the lab safely and be safe. There are some nasty things that you are going to use in this lab. So we do have you read and sign a safety consent form in the experiment 1 folder before you attend the first lab. That form goes over a lot of the general policies that we have about safety. So that's basically that. So I want to briefly go over safety. Safety is the most important thing, and we spend a lot more time on this, but this is a video, and I know that if the videos get too long, it's difficult to get people to watch them. So I want you to just, be reading that safety consent form carefully, especially if you missed the first lab. So briefly, wear goggles at all times when anyone is doing chemistry in the lab. This is absolutely required by law. It's also for your own good. If you get some of these things in your eyes, you're going to have big problems. So it's really important that you're wearing goggles at all times. If you get something on yourself, if you spill something on your hand, wash it off thoroughly and immediately. Okay. Basically, you want to rinse it off with water. The single best thing you can do is get it off of yourself quickly. Some of this stuff will cause chemical burns, and it's really important that you get it off quickly. Along these lines, you should be wearing gloves when you're working with these chemicals to protect your hands, because obviously that's the most likely place that you're going to spill something, but it's not the only possible place to spill something. So just be aware of that. It's also important to note that you do wear gloves when you're working with chemicals, but if you're using the keyboard, if you go in the hall, you don't wear gloves, because if you get something on your hands and you put it on the door handle or the keyboard, and then the next person isn't wearing gloves when they use the keyboard or the door handle, then they can get it on them, and we don't want that to happen. Also, just as a general rule, if you spill something, clean it up, okay? Just wipe it up. You know what it is. Maybe it's just salt and it can go into the garbage can, or maybe it's some hazardous chemical and it needs to be cleaned up more carefully. Of course, you have a TA in there that can always help you, and you can ask and should ask if something happens. You spill something on yourself. But if you spill something on yourself, don't wait to ask if the TA is busy. Go rinse it off and then tell the TA. Kind of as you're rinsing it off, you'll see if you haven't been in there, they're pretty small rooms, so it's pretty easy to get someone's attention. Okay, fire alarm. If the fire alarm sounds, you definitely don't want to be in the chemistry building. There's all kinds of nasty stuff in this building because people are doing chemistry, both teaching-wise and research, so you don't want to be in here. So please exit the building immediately. So just turn off what you're doing, exit the building immediately, and meet in the courtyard between the chemistry and biology to await further notice. Sometimes the alarm goes off for 5 minutes. Sometimes it goes off for hours. So we'll do our best to let you know what to do as far as should you continue to wait to go back into the building. And sometimes we don't know, but we'll do our best to get you information. Okay, and then of course no food or drink in the lab. It is a chemistry lab. Please don't bring Starbucks that you just spend a bunch of money for to the lab with you. That's not a good idea because we can't let you have it in the lab, and then it'll be cold or warm depending on what you get by the time you can get to it. So you can't bring it into the lab. So as a continuation of safety, we do have a dress code in the lab. So generally speaking, it is completely covered legs and feet. No shorts or skirts that don't completely cover your legs. No sandals, no Crocs, no shoes with a bunch of holes in them. Okay, so completely covered legs and feet. And of course, this is for your own safety. Because of gravity, if you spill something, it will go down. Okay, and you don't want it to go down onto your legs and your feet. You'd much rather go onto your pants, even though it might ruin your favorite pair of pants. That's a lot better than getting a chemical burn on your skin. If you have long hair, do tie it back. All right, you don't want it to dangle into the flasks and things like this, and then no dangling jewelry as well. So all of these things are mainly just for your own safety. These are normal lab things that people are doing in all labs everywhere, and they're things you just need to get used to doing. I was over at Regeneron recently, and the people were all doing all of these safety rules there as well, because there's dangerous stuff and we don't want anyone to get hurt, and we don't want you guys to get hurt. So please just follow the rules. So now what I'm going to do is kind of change over this video and go over to the Brightspace page, show you a little bit about that, and show you a little bit about the first pre-lab. So this is the Brightspace page. All right, you're in ACHM 115 if you're watching this video, so you can see the weekly schedule, and the weekly schedule will tell you, what experiment we're doing. So depending on when you're watching this, this might be what you're doing next week. This may not even be made in the semester you're taking this course, because this video should hopefully last for a while. The quiz you're taking, the weekly announcement will tell you what's on the quiz, and it will tell you homework due dates as well. So what homework is due that week. So Monday, August 28th for this particular semester, this is what's due, and it'll tell you when the exam is. So that's basically a very important thing to be aware of, is the course calendar. The lecture, I'm going to leave that. Dr. Siahi will talk more about that. Homework, so this is where you can find your homework. Sometimes it can be easier to go in through the content folder and go through the homework. And if we look at chapter 1, you'll see that there's a few things that are in here. Okay, you're going to get the these are just some tips about how to practice for general chemistry. It's super important. The vocabulary, words that are on the homework. There are not graded practice problems, which is going to include the supported practice problems and the unsupported practice problems. And the supported practice problems have videos. That's what distinguishes them from the unsupported practice problems. So here's the unsupported, here are the supported. You also have the worked out answers for the supported practice problems. So these are just practice problems like you'd find in the back of any textbook. All right, they're a good idea to do, but as you can clearly see here, they're not graded. And there is a video playlist, which will just take you over to YouTube. All right, and there's a bunch of boring videos that you can watch where I go over different problems. Okay, so that's basically that. Now I need to go back to chapter 1. Okay, and this is again in the content folder. All right, and then finally you'll find the randomized online homework. So the randomized online homework is like a homework quiz. You get 5 attempts. Your best score counts. So it's kind of a quiz, but it's more homework. So that is basically that, and it has a due date, okay, and it has an availability date. So just be aware of that. There is a time when it ends, and you can no longer look at it. All right, so just be aware of that. Make sure you do it before the due date. The other thing is you have this Chapter 1 online homework equivalency table, and if you click on this, it basically, if you're stuck on question 6, you might want to watch this video. And these are from the supported practice problems. It's a similar problem that goes over a similar concept, and you can watch the video and you can find out how to see an example of that problem. So going back to the tour of the Brightspace, so that's the homework. Now we can go to the labs section. Okay, so for the lab section, you can find the syllabus. This is the safety consent form that you need to go ahead and sign before your first lab. The lab makeup policy, we do not excuse any labs, so just be aware of that and make sure you take a look at this policy. There are opportunities to make up missed labs, but we do not excuse any labs. And then the different experiments throughout the semester. Right now we're on experiment 1. So what you'll see here for the first 3 experiments, you'll see the experiment 1 excerpt. This is just from the lab manual. We literally just took it out of the lab manual and put it up here on Brightspace. So that is basically that. You can see that there is an experiment 1 video, which talks about what we're doing in experiment 1. There's the first lab checklist. So this is the things you need to have for the very first day. And these are things that we just covered a few minutes ago. Okay, safety consent form, proper personal protective equipment, dressed appropriately for lab, completely covered legs and feet, and you're ready to go with your Excel file. I'll show that in a minute. You've completed your first pre-lab quiz, and you're prepared to take the Chapter 1 quiz. So these are basically just a checklist just to make sure that you're prepared for your very first lab. Then we have the pre-lab quiz and the experiment 1 Excel file. So next, I'd like to talk about the pre-lab quiz in a little bit more detail. All right, so I'm going to go ahead and click on this pre-lab quiz, and I'm going to click on preview. You won't have to do this. You'll just have to go to start quiz. All right, and we can see this quiz. So this quiz will give you 5 attempts. So this is just letting you know that it has a density exercise, which is not covered in the pre-lab quiz, but it is something that you will do in the lab. So if you go to the experiment 1 lab manual expert or the experiment 1 in the lab manual, they're exactly the same. It'll talk about that density exercise. You're basically going to do density of a metal by displacement. You're going to put some in water, and it's going to displace the water. You're going to find the volume by displacement. You're going to find the mass by measuring the mass on the balance, this kind of stuff. The objective of the experiment, so the pre-lab is broken into several parts: the objective, the hypothesis, the variables, the experimental procedure, and the chemical hazards and waste. And you're writing your pre-lab kind of in the form of a quiz. And this is a low stakes quiz. All right, you get 5 attempts. You only have to redo the problems that you got wrong. Unlike the homework quiz. The homework quiz, if you want to do another attempt to get a higher grade, you have to redo all the problems, because in chemistry you have to practice. We want to incentivize you to practice because practice makes perfect. Okay, when I took this class, I had to practice a lot. So it'll ask you for the objective of the experiment. Once you've read the experiment, this should be fairly you should be able to determine this. All right, the hypothesis, this may be a little bit more challenging, but you can just what type of solution that seawater is, and you can look here and look at the different answers. Okay, seawater is a homogeneous mixture. The mass of salt in 5 milliliters of 3% M over V salt water. Okay, this is a dimensional analysis problem, so you want to convert 5 milliliters of solution to mass of salt, and you're going to find out that it's 0.15 grams. And how the salt will be isolated. So this is basically a hypothesis. What do you expect to find? 0.15 grams of salt. Why do you find that? Because salt water is a homogeneous mixture, so it doesn't matter which 5 milliliters you take out of the 25 milliliters you make, you're going to get the same amount of salt in each one. And then how the salt will be isolated, which I'm not going to answer all of these. Okay, the independent and the dependent variable. The independent variable is the experimenter controlled variable. What does the experimenter control? And the dependent variable is the variable that you are actually measuring. So in this case, you're trying to find the mass of salt in the 5 milliliter aliquot. So that is basically the dependent variable. The independent variable is the volume of the aliquot. Okay, how much of the aliquot did you take? You decided to take, well, I guess we decided that you would take 5 milliliters, okay? But then you want to find the mass of the salt. So what are you looking for as the dependent variable? What are you controlling as the independent variable? All right, the melting point of salt isn't really a variable, and the temperature of the hot plate doesn't really play a strong role here because you're boiling off the water, so it's not really a variable either, because you could argue that it's a variable, but it doesn't really matter. Okay, so basically that are your independent and your dependent variable. Next, you're going to do the experimental procedure. So the experimental procedure is the experimental steps in proper order. So this is the steps to do the experiment. These are written in the lab manual, but not step by step, because there's some theory in there of why you're doing things in the order and why you're doing the things you're doing. All right, we don't want you to just be able to do this experiment. We want you to understand how chemists think about these types of problems, because this is what scientists need to be able to do. So we're trying to give you some experience with that. So you can go ahead and just place these in order. What are you going to do first? What are you going to do second? So on and so forth. Again, it's 5 attempts. This could be a little bit challenging. You might get this wrong the first time. If you get it wrong, you'll get another chance to do it. So you can basically do that. Then there's some safety questions, like what do you need to wear in the lab at all times? Goggles. You need to definitely wear goggles. You need to wear a lab coat, and you need to wear gloves when actively working with harmful chemicals. Now, if you're using salt water, do you necessarily need to wear gloves? No. If you've ever been in the ocean, you've been covered in salt water, and you were probably totally fine. So, if it's salt water, you don't necessarily need to wear gloves. If ever in doubt, you can refer to the SDS sheet, which tells you the chemical safety of the things that you're working with, or you could talk to the TA or me or Jesse. You can send us an email if you're ever in doubt about whether you should wear gloves or not. Just ask somebody or refer to the SDS sheet. All right, potential hazards of the experiment. The main thing here is you're using hot plates. Hot plates are hot. It's like using a stove, okay? And they melt electrical cords. This is actually a common problem, okay? They can melt electrical cords, they can melt mouse cords and keyboard cords, and all other kinds of cords. So just make sure there's no cords on them, okay? And the flasks do tend to splatter, so don't stick your face right over the flask, because it could splatter into your face, not your eye, because you're wearing goggles. But that's basically that. Chemical waste, we're just using salt water, so that is basically can go down the drain with some water to flush it down so that it doesn't leave a salty mess in the sink. All right, so that is basically that. The last part of the quiz is going to be the data analysis part. Okay, so this is basically some data that's made up. It's ridiculous because the mass of the flask is going to end up being like 400 grams, or is it 400 grams? These flasks probably weigh 20 grams, not 400. It is made up, but it's an opportunity for you to analyze the data before you actually come to lab. Again, this is designed so you get multiple chances. If you get something wrong, you will get another chance to do it, okay? And basically you'll only have to redo the ones that you didn't do. One last thing I want to say before I go over the data analysis, I'm going to spend a little bit more time on this, is unfortunately to get out of the quiz, I have to submit quiz even though I didn't answer anything, so I'll come back into it in a minute. When you get out of here, if you go to the content area, and unfortunately you have to dig back down in with Brightspace, and you go to experiment 1, what you're going to find is that you're going to have the experiment 1 pre-lab. So there's 2 conditions you need to meet in order to do this. You need to either get full credit on the pre-lab quiz, which means you get them all right. Again, you could redo the ones you don't know how to that you get wrong, okay? Or you've extinguished all 5 of your attempts. Then you'll get the experiment 1 pre-lab, and you'll notice the experiment 1 pre-lab is the answers to all the things. Okay, the title is just the same as the title in the lab manual, but the objective of the experiment that you answered a question about, the hypothesis that you answered several questions about, the experimental procedure that you put in order. Now it's nicely placed in order for you. The variables, okay, the dependent variable, the mass of the salt, and the independent, the volume of the aliquot, and the chemical hazards and waste, which was mainly in this case was the hot plate and the salt water can go down the drain. So that's basically that. Also, the flask does splatter, so don't stick your face right over it. So this you can then print. Again, you don't have to print it. You can view it on Brightspace, and there are computers in the lab. So basically, you have this when you actually do the lab next week. What you also need is the data analysis. So I want to spend the rest of this video talking about the data analysis. All right, so for the experiment one data analysis, before I go ahead and get into the quiz, I want to do the experiment one Excel file. So if you click on that, you can see this Excel file, then you want to download it. And I do want to warn you that all videos for the entire semester are made using the PC version of Excel. My understanding is that the Mac version will do all the same things as a PC version, but you might have to click different buttons. Why they make it like that, I couldn't tell you, okay? But just be aware that we don't have Mac computers, so we're not good at helping with Mac computers. So there are PCs in the lab, all right, and the videos will go kind of along with the lab. Or, you know, if you're not sure how to do something, often it's probably similar, and a Google search could probably solve your problem. So this is the experiment one data analysis file. So what we're doing in experiment one is we're taking and making 25 milliliters of salt water. All right, after we make the 25 milliliters of salt water solution, what we're going to do is take 3 5 milliliter aliquots, put it into masked 50 milliliter Erlenmeyer flasks, and boil off the water on a hot plate. You've probably gotten most of that from the quiz questions, and I probably should have said that before I went over the quiz. That being said, what you're going to have is 2 masses for each trial. So there are 3 trials, and you're going to have 2 masses. The mass of the empty flask before you put the salt water in it, and then the mass of the residual salt in the flask. So you're going to put 5 milliliters in it, you're going to put it on the hot plate, you're going to boil off the water, and the salt will remain. You'll literally see the salt in the flask. So there'll be some salt in the bottom of the flask. Then you'll weigh it again, and you'll get a new mass. This mass should be 0.15 grams higher than this mass. Why? Because you're boiling off 5 milliliters of salt water, and it's 3% solution, so it should have 0.15 grams of salt in it. All right, so that is basically how this works. We're then going to talk about the accuracy and the precision. But before we do that, what I want to do is copy and paste in a couple things here that you don't have. So now you can see I've pasted in this bullseye and a couple of formulas. So first thing we want to do is talk a little bit about what are accuracy and precision, what do they mean, and then we'll talk about how to calculate them. Okay, so first you can see this is some type of darts or arrows or bullets or whatever it is shooting at the bullseye. All right, and you can see that these 3 or 4 shots are all close to the bullseye, all right, and close to each other. These are both accurate and precise. Note that we're talking about this in terms of a bullseye, but we can also talk about this in terms of data. We do a bunch of trials, and all of our results come out to be the same. All right, so they're clustered, and they're close to what we expect. So they're close to the bullseye. In this case, we want 0.15 grams of salt in each trial. That is going to be reproducible data if all 3 trials give us the same amount. And we expect 0.15 grams, so it's also close to the bullseye. We could have this, all right? We're trying to hit here. We take 4 shots, and we're all over the place. Not only are these not close together, they're also not close to the bullseye. So this is kind of like you get random numbers. You do this 4 times, and instead of getting 0.15 grams, you get 1 gram and 0.01 gram and 0.2 grams and 0.4 grams. So you can't get the same thing every time, and it's not close to the 0.15 grams that you expect. So this is not accurate and not precise. Now here's an interesting case. Here's an interesting case where the shots are clustered. They're close together, but they're not close to the bullseye, or they're not close to what you expect. That would be for the salt, like getting 0.5 grams, but getting it 3 times in a row. So it's 0.5 grams, 0.5 grams, 0.5 grams. It's very close together, but it is not very close to what we expect. And in this case, it means there's some type of systematic error, some like with the balance. In research data, sometimes it means that your bullseye is actually over here. You're gonna have to do a lot more work to try to prove that, but maybe your bullseye is actually over here, and what you expected is actually the wrong expectation. That can happen as well. But in this case, the expectation is pretty solid, okay, if the solution was made properly, but maybe there's something wrong with the balance, or maybe the solution was made wrong to begin with, so it wasn't actually a 3% solution. So those are 2 possibilities to get this type of data. At the end of the day, what we need to know is this data is precise, it's reproducible, but it's not accurate, it's not close to the bullseye. So again, accurate and precise. Accurate because it's close to the bullseye, precise because it's reproducible. Not accurate and not precise. Not accurate because none of the shots are close to the bullseye. Not precise because they're also not grouped. They're not close to each other. This data, not accurate because it's not close to the bullseye, yet precise because it's reproducible. In order to test the precision of any type of data, you need to do multiple trials because precision is how close are the trials to each other. In order to test the accuracy of any type of data, you need to know what the accepted value is. In this case, our accepted value is 0.15 grams. So, what do we calculate in order to do this? What we calculate to test precision is the relative standard deviation, which is the standard deviation divided by the average times 1,000. So the standard deviation, which is the typical thing you use to test precision, is relative because it's divided by the average, and then we multiply it by 1,000 just to make it a nice number. Okay. The accuracy is tested by the percent error, which is the experimental value. We have used the average, so you only have to calculate it once, minus the accepted value divided by the accepted value times 100. In this case, it's the average experimental value, which we'll calculate in a minute, divided by the accepted value, 0.15 grams, divided by 0.15 grams times 100 because it's a percentage. That's the percent error from the accepted value. So this is basically how we do this. Now, what you need to know is how do you actually calculate this in Excel? So what I'm going to do is go back into the experiment 1 pre-lab quiz. Again, you don't have to hit preview, okay? And I'm going to hit start quiz here. All right, and what you'll find is you have this data. So it says a student performed experiment 1 by evaporating 5 milliliters of a 3% NaCl solution. You're going to be this student when you do this experiment. The data below is what they obtained. Calculate the average mass of the salt remaining in grams. That's the first 4 answers. The relative standard deviation in parts per thousand, that's the second 4 answers, and the percent error, the third 4 answers. So that's basically how you have to do this. Now, what you want to first do is take and copy this data, and you'll notice it's red and blue, and you'll also notice that on the Excel file, okay, you have red and blue. So the red data goes where the red data does, and the blue goes where the blue goes. So when you do the copy and paste, you don't want to use the shortcut key Control C, Control V. If you do that, for some reason, it just pastes it as text. But if you highlight the data, and you right click and you hit copy, and then you come over to your data and you right click and you hit paste, then it seems to copy and paste in. Note that these data are nonsense. There's no flask, 50 milliliter Erlenmeyer flask, that weighs 436 grams. 454 grams is a pound. All right, that's a pretty heavy flask. All right, it would have to be a very large flask for that to be the case. 50 milliliter flask is basically the size of a shot glass, so it's very small. Okay, so that's basically that. It doesn't look like a shot glass, it's just about the size of a shot glass. So now we need to find the mass of salt for each trial. That's to find the individual data points because we don't want the flask. We have the flask and the residual salt. So we want to subtract out the mass of the flask to find the mass of the salt. To do that, we have to tell Excel to do math. You must hit equals to tell Excel to do math. Then you take the mass of the flask and the residual salt, simply click on it, minus, which is just the minus key, and the mass of the flask. This value, again, you simply click on it, hit enter. As a sanity check, it should be around 0.15 grams, and it is right around 0.15 grams. Now I'd like to do that for all 3 trials. I simply hover my mouse over this little green square, this green square, until I see this plus sign. So you'll see this arrow change from this kind of plus to this kind of plus. When I see this kind of plus, I can just click my mouse button and drag it down. Now I have the masses for all 3 trials. It is absolutely required that you have this as a math formula. You cannot subtract these numbers by hand and just type in. We're going to check to make sure you didn't do that. You have to use Excel to do the math because part of this course is learning how to use Excel to do math. In this case, we have 3 trials. When you're doing scientific work with instruments, it's not uncommon to have 1,000 data points. If you have 1,000 data points, you are not going to manipulate them by hand. Nobody's doing that because it would take you so long to do 1,000 subtractions, and you can just type it in once and drag it down if you're doing it with Excel. Now I want to calculate the average. Well, it turns out that Excel knows how to calculate the average. Equals average, just the word average. Open a parenthesis. When you have a formula that isn't simply just with the keys plus, minus, multiply, divide, if you have a formula like average, you have to open a parenthesis. Then you simply highlight your data. So I'm just clicking and dragging down, and now I'm going to take the average of H4. So this is H4 through H6. Okay, now you can either close the parentheses with the keyboard. You don't have to. You can just hit enter, and you'll notice if you go back, it automatically knows to close the parentheses. Okay, now the standard deviation in grams. So we need to find the standard deviation, because remember, we're trying to find the relative standard deviation in parts per thousand, which is the standard deviation divided by the average times 1,000, which is a measure of precision. How close are the data together? If we look at this data is clearly close together. Okay, so part of determining whether it's close together or these data all about the same is calculating the standard deviation. To do that, you tell Excel to do it for you. Excel's formula is STDEV. Open a parenthesis. Select your data. Again, H4 to H6. Do not include the average. All right, it's very important that you don't include the average here. You only want to find the standard deviation of your data points, which is how close they are together. We close the parentheses or not, and then hit enter. Okay, and what you'll notice is the standard deviation is very low. The best standard deviation is 0. In order to get a standard deviation of 0, these numbers would all have to be exactly the same. They're not all exactly the same, so it's not quite 0, but it's very close. All right, this is very precise data. If you work carefully in the lab, you can get precise data. Will it be this low? I don't know. All right, but it'll be, low. Now we want to find the relative standard deviation, which again is the standard deviation divided by the average times 1,000. Well, you just calculated the standard deviation, so just click it. Divide is the slash. Okay, if you have the number pad, you can find the divide on there, or you can find it on the regular part of the keyboard. The average, so I just click it, times 1,000. Okay, so I just multiply that by 1,000, and I find the relative standard deviation. And you'll see it's 11 out of 1,000, which is 0.1%. which is meaning that our data is fairly precise, meaning that it's grouped. All right, we don't know if it's grouped like this or grouped like this until we calculate the percent error. But remember that the accepted value here is 0.15 grams and our average value is 0.15 grams. Now, oftentimes people will calculate the percent error of every data point. But for this class, we're not going to make you do that. You can calculate the percent error of the average. That can be misleading because you could have very, very imprecise data where the average just by some coincidence happens to be in the middle, and you might say it's accurate. So you have to be careful doing that. That being said, it is easier to do it that way because you only have to do it once. So that's basically how we're going to have you do it. So the percent error is going to be equal to the absolute value of the experimental value, in this case our average experimental value, minus the accepted value, 0.15 grams, divided by the accepted value, 0.515 grams, times 100. Again, we can see our average is very close to 0.15 grams, so we know our percent error is low. To have Excel do the absolute value, we type in ABS, open a parenthesis. Okay, we want the experimental average, which is the one we just found, minus the accepted value, 0.150. Okay, this 0 you don't actually need because Excel will delete it, but that's fine. Divided by the accepted value, 0.15 times 100. So we have the absolute value of the experimental value, which is in cell H38, minus the experimental value minus the accepted value, 0.15, divided by the accepted value, 0.15, again 0.15 or 0.150, Excel will interpret them the same, times 100. All right, so that's basically what we did, and we can see that our percent error is 0.2%. So we can say this data is precise because the RSD is low, the relative standard deviation is low, and this data is accurate, again, because the percent error is low. This tells us the precision, how close and reproducible the data is, how close they are to each other, and this tells us the accuracy, how close they are to the accepted value. And these are basically the calculations that we're doing in the lab. So the final thing that I want to tell you is that this Excel file is very valuable. So if you go here, you can see that these answers are all here. So it asks us, calculate the average mass of salt remaining, which is 0.1503 grams, which is right there. Note that the sig figs in Excel, it's like a calculator. It just calculates a whole bunch of sig figs, and the code to fix sig figs is very long and computer programming. So we're not going to ask you to fix the sig figs. All right. Then it asks us for the relative standard deviation in parts per thousand, which is 11.37. Okay, and to just the first two digits, it's 11. All right, and then it asks us to find the percent error, which is 0.2, and it's right there as 0.200. So you could see that we have all those answers. This is really important because this means, and when you submit this, you'll get it correct, this means that this data file is analyzed correctly. That means that this data file is very valuable. You are going to want to save this data file because if I delete this data and then I put in my actual data when I go to the lab, it will automatically, that should be at 20, it will automatically do the calculations for me. And when I put in my 3 data points, it will now calculate it. And I know that this is all calculated correctly because I already tested it. So it's really a good idea to save your data file so that you don't have to type in all of the numbers a second time. All right, I hope that you found this video helpful, and I hope that it helps you to understand what's going on in lab. In the lab, they spend about 2 hours, so you get all of this in about 35 minutes. It's still long, I get that, but it is better than 2 hours. Thanks for watching.